Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th students, 11th biology students, and this is meant for 19th November 2020. As all of you know, we are into the revision mode and we are revising chapter number 9, biomolecules, and in that biomolecules we were talking about the enzymes. And in our previous session, if you recollect from your memory, we said factors affecting enzymes, and in that we said three factors will affect the enzymes. Like one, we said temperature. The first factor that influences the enzymatic activity is the temperature, and it has to be optimum temperature. Anything more or less the temperature, it will denature the enzyme. And second, we said pH. Again, it has to be optimum pH, and the optimum pH is neutral pH, that is 7. And you are seeing in the graphical form that when temperature or pH on the extremes, the enzymes get denatured, and when it is in the optimum, the enzymatic activity reaches the peak. And then third we discussed is concentration of the substrate. So more the concentration, the faster this. So increase in substrate concentration increases the rate of reaction due to occupation of more active sites by the substrate. Then competitive inhibitor. When the molecular structure of inhibitor resembles the substrate, it inhibits, means does not encourage. It suppresses the function of enzymes. Now today we are entering into classification of enzymes. Basically, as all of you know, an enzyme is a protein, as I said in our previous session. It can be an RNA produced by living cells, which is highly specific and highly catalytic to its substrates. Enzymes are very important type of macromolecular biological catalysts. Macro, big molecules, big in size. Due to the action of enzymes, chemical reactions in organisms can also be carried out efficiently and specifically under mild conditions. And when we go for the nomenclature, how do you name an enzyme? As we discussed previously, anything is enzyme, the name will end up with ASE, ASE, right? Like oxidoreductase, dehydrogenase, transferase. Look at that, ASE. So the nomenclature of enzymes is derived from their substrate or the catalyzed chemical reactions, and ASE is usually added as suffix. Enzymes can be indexed with letters and numbers according to International Union of Biochemistry and Molecular Biology. The letter EC plus 4 numbers representing 4 elements. The first number represents enzymes that are classified according to the mechanism of enzymatic action. So how do you classify the enzymes? According to the type of reactions that the enzymes catalyze, enzymes are classified into 7 categories, which are oxidoreductases or dehydrogenases. And the second one, after oxidoreductase, it is transferases. After transferases, it is hydrolases. Third one, hydrolases. And then ligases. And the fourth, isomerase. Fifth. And the sixth, ligases, and translocases. So there is a seventh one, translocases. So oxidoreductases, transferases, and transferases and hydrolases are the most abundant forms of enzymes. Individual enzyme classes are further classified systematically based on the chemical name of the substrate and its reaction mechanism. Example, first one, oxidoreductases. So in oxidoreductases, the type of reaction is substrate A, for example, it is reduction, it is getting reduced, and B is getting oxidized. So when you get this product, the A will be oxido and B will be reductase. That means it is getting catalyzed redox reaction and can be categorized into oxidase as well as reductase. So one is getting oxidized, another is getting reduced in the presence of enzyme. That's why we call that such enzymes as oxidoreductases. And in the process, there's a removal of dehydrogenase. Hydrogen is removed. That's why the name dehydrogenase. So S is reduced and another S is getting oxidized. Substrate 1, substrate 2. And what is happening? S is getting oxidized, product S dash is reduced. And then we come to the second one, transferases. In transferases, we can write the reaction in this form. A minus B plus C giving rise to the products A plus BC. So here it catalyzes the transfer or exchange of certain groups among the substrates. So there is exchange of groups. As you are seeing here, the C is free in the reactant side, and the product side it is B and C is joined together, A is getting separated. So it catalyzes the transfer or exchange of certain groups among some substrates. And the third one, hydrolases. In hydrolases, the reaction can be written as, I can write here, A dash B plus H2O, hydrolases, forming A dash H and B dash OH. So here what's happening? Accelerate the hydrolysis of substrates. Hydros means water, lysis means breaking. So the H2O is getting broken into H plus and OH minus. So that's how the hydrolases act. So they catalyze the hydrolysis of peptide, ester, glycosidic bonds, etc. And that is the function of hydrolases. And now all important ligases. Many times we discussed this. That is reverse reaction synthase. Like here you can see C and X minus Y. Here you can see the ligases being formed. Ligases. In the ligases, A dash B, it is reversible. And the reversible, it becomes A plus B. So here, reverse reaction. It promotes the removal of a group from the substrate to leave a double bond reaction or catalyze its reverse reaction. So this is the function. So first it promotes the removal of a group. So there is a removal of a group from the substrate to leave a double bond reaction. You can see here there is a double bond and it catalyzes reverse reaction. That's how ligases function. And then isomerases. Isomerases basically interconversion of optical and geometrical or positional isomers. You may recollect from chemistry knowledge what are isomers, and we can write this in this form. The mirror images A dash B dash C, this reversible reaction, and what you get is A dash C dash B. Now here in the center C, here in the center it's B. So facilitates the conversion of isomers, geometric isomers or optical isomers. Basically isomers, of course you know, they are mirror images. So isomerases are the enzymes that help in formation of the isomers, and the isoisomers geometric isomers and optical isomers are being formed. And then the one that we have been discussing in enzymes, very important one, ligases. Ligases, of course, they catalyzes the linking together of two compounds. So the compounds, we can write the reaction A plus B. Of course, there is involvement of ATP molecules. Energy is spent to link together, and this forms A dash B with ATP being spent. ATP splits into adenosine triphosphate, splits into adenosine diphosphate plus inorganic phosphate. So catalyzes the synthesis of two molecular substances into one molecular compound. So this is individual substances, two molecular substances, and it is making one joint compound, molecular compound, with the release of energy. ATPs are spent. That's why it is ADP and inorganic phosphate. And the next one is the translocases. So seventh one also you can note down. It is translocases. So in translocases, it catalyzes the movement of ions. Ions are molecules across membranes or their separation within the membranes. So it is the cross transfer of ions or molecules across the membranes. According to the Unified Classification Principle of Enzymes published by the International Society of Biochemistry, each group of enzymes in the above 7 categories can be further divided into several subgroups according to the characteristics of functional groups or bonds in the substrates. In order to show the properties of substrates or reactants more accurately, each subclass is further divided into subclasses and directly contains a quantity of enzymes. Moreover, on the basis of molecular composition, enzymes can be divided into pure enzymes and binding enzymes. Enzymes containing only protein are called pure enzymes, and the binding enzymes are composed of proteins and cofactors. Look at that. So it's the binding enzymes that are composed of proteins and cofactors. Only when the two components are combined can the enzyme have catalytic activity. So what are cofactors? We said basically enzymes are proteins, but cofactors are non-protein. That's the difference. Non-protein constituent of an enzyme, which make the enzyme more catalytically active. It helps in driving the catalysis faster, catalytic faster. The protein portions of the enzyme are called apoenzymes. So cofactors are having this. The prosthetic groups, that is organic compounds, coenzymes, organic compounds with the transit association, and of course metal ions and zinc, etc. So the prosthetic groups are organic compounds. See, when we say organic compounds, it is carbon is involved. Carbonated compounds are involved. So prosthetic groups are organic compounds and are tightly bound to apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide, heme is the prosthetic group. The essential chemical components of any coenzymes are vitamins. Example, coenzyme NAD, nicotinamide adenine diphosphate. NAD and NADP phosphate contain vitamin niacin. So this is how the enzymes are classified, and there is a combination of protein as well as non-protein, which is called cofactor. And every enzyme having a link with the substrate, and we call that mechanism as lock and key mechanism. If you recollect from your memory, lock and key mechanism. It is just like the lock will open only when a correct key is inserted. The key, as you must have observed, even in our day-to-day life, will have teeth or will have rows or the compartments. So the key having the teeth or the rows should enter inside the lock and fix it. Then only lock and key gets in coordination and lock gets opened. Same is the case, the enzyme and substrate both should function as lock and key. Then only you get what is called ES complex, enzyme substrate complex. So basically if we analyze, any enzyme will have first substrate. So this substrate plus E for enzyme, and when reaction takes place, in between you have got substrate enzyme complex, and the enzyme helps in converting the substrate into a product, and this product is of very useful, and the enzyme comes off, enzyme gets separated. As I said, if you recollect from your memory, I said enzyme will not undergo any reaction itself. It's just like a cricket umpire, I said. Just like how the cricket umpire will not do batting, will not do bowling, will not do fielding also, but he will ensure that the game goes on smoothly without any problem. And the match gets started at a particular time and gets over at a particular time. You must have also noticed that umpires putting fine on a particular player who is wasting the time, or on a captain whose team is delaying or following the delaying tactics, so that the probable victory can be stopped or can be nullified. So enzymes are exactly like that. They do not take part directly in the reaction, but ensure that the substrate quickly becomes the product. Or we can also call this substrate sometimes reactant. So reactants react together and form products. And how quickly these products are formed from the reactants depends upon the enzyme. So enzyme ensures that the rate of reaction at which the substrate gets converted into product is minimum. So the speed will be faster and the time is minimum. This is how we said that some of the factors that influence the enzymatic activity, we said concentration of the substrate. It increases the substrate concentration, increases the rate of reaction. So that's important. So you can see the graphical representation also in the same way, pH, time, as well as concentration of substance. More the concentration, faster the reaction. So this is how the enzymes work. Keep practicing, draw the diagrams, draw the graphs, and just to see on x-axis what you are plotting and y-axis what you are plotting, and how the graph gets into the picture. And sometimes you may have to think out of box, like if the graph is this is the normal graph, but in your exam you may get the graph in reverse side, and he will ask you interpret this graph, why this happens, when this happens, and how to do the corrective measures. So this is interpretation question. Such questions are very common, and these are called HOTS, high order thinking skills. So you need to practice such questions also. So we will give a lot of questions in our WhatsApp group. Keep